We are going to explain affinity maturation. Premise: B cells produce antibodies, immunoglobulins, in response to antigens. This antibody protects the body from antigens by binding to them and neutralizing them. It is an important component of acquired immunity and humoral immunity. Definition: affinity maturation is a phenomenon in which antibodies that bind more easily to antigens, that is, antibodies with higher antigen affinity, are produced as time passes after immunization or as immunization repeated over and over. Place: affinity maturation occurs in germinal centers. Germinal centers are microstructures that form in secondary immune tissues such as the spleen and lymph nodes. There are two types of regions in the germinal center: dark regions and light regions. B cells first proliferate in the dark region, then migrate to the light region, where they are selected and differentiate into memory B cells and plasma cells. Affinity maturation occurs during this process. There is a two-step mechanism for affinity maturation. Somatic hypermutation occurs in the dark region, and high affinity B cell selection occurs in the light region. The first process is somatic hypermutation. As B cells proliferate, point mutations are highly likely to occur in the region of the gene that encodes immunoglobulins. This mutation usually changes the region of the immunoglobulin that bind to the antigen, so it affects antigen binding affinity of antibodies. Patterns of point mutations are highly variable. This diversity gives rise to a variety of B cells with antibodies with high to low antigen binding affinities. Among such a variety of B cells, there is a mechanism by which B cells with antibodies with higher affinities are selected and survive. Follicular dendritic cells and follicular helper T cells play an important role here. Follicular dendritic cells retain antigens on the cell surface for a long time and efficiently present antigens to B cells. Antibodies act as receptors on the surface of B cells and receive the presented antigen into the B cells. At this time, the higher the antigen affinity of the antibody, the easier it is to receive the antigen. The antigen is processed in the cells and then presented to follicular helper T cells, which then give survival signal to the B cells. B cells that do not receive the survival signal undergo apoptosis and are eliminated. Therefore, B cells that can receive survival signals more efficiently survive. So B cells that produce antibodies with high antigen affinity survive. As affinity maturation progresses, high affinity antibodies increase. Then antigens in the body are effectively neutralized and reduced. Antigens are a necessary resource for B cells to receive survival signals, as explained in the last slide. Therefore, the fewer antigens there are, the more competition there is for those resources, and higher affinity is required for survival. As a result, affinity maturation increasingly progresses. Affinity maturation is an important system for effectively neutralizing pathogens.